Here's the latest audio research news from the transmitter. Neurons fuel lung tumors that have spread to brain, by Sarah D. Weir. Neurons in the brain form functional connections with small cell lung cancer cells that have spread there, fueling the growth of these brain metastases. Multiple experiments by two independent teams of researchers underpin the new finding. The development adds to mounting evidence of a role for the nervous system in cancer initiation and progression. Neurons form synapses with brain cancer cells, past studies show, and the nervous system is also known to regulate the growth of many cancers outside the central nervous system. But the new work provides the first definitive demonstration of functional synapses between neurons and cancer cells that originate outside the central nervous system. It really pushes cancer neuroscience to the next level, says Hamsa Venkatesh, assistant professor of neurology at Harvard Medical School and an investigator on one of the studies. The studies, both published September 10th in Nature, each involve dozens of interdisciplinary researchers. One group, which includes Venkatesh, is based in the US and the other in Germany. Together, they marshal evidence from gene expression, electron microscopy, cell culture, electrophysiology, and optogenetics, and draw on a variety of other experiments as well. I loved how they used some similar techniques, but other very different techniques, and came to the very same conclusion, says Paula Vermeer, associate professor of surgery at the University of South Dakota, who was not involved in either effort. It's just a tour de force on both sides. Small cell lung cancer often spreads to the brain, and tissue samples from people with brain metastases show axons in and around these tumors. A similar pattern of axon growth occurs in the brains of mice injected with small cell lung cancer cells that form tumors there, both teams showed. Viewed under powerful microscopes at nanoscale resolution, the contacts between the nerve cells and cancer cells in the mouse brains look like typical synapses between neurons, both teams observed. And the synapses are functional, electrical current recordings from individual cancer cells in brain slices showed, providing compelling evidence of synaptic transmission. In addition, a fluorescent dye moved from cancer cell to neuron in the brain, according to viral tracing studies by the German team. That team has also documented synaptic structures in primary lung tumors in mice and in laboratory dishes where neurons are cultured alongside small cell lung cancer cells. The lab dish synapses are functional, but it is not yet certain whether the lung tumor synapses are. We're working on that, says study investigator Filippo Bellega, a cancer geneticist at the University of Cologne. That's very high on our list of priorities. Neuronal input to small cell lung cancer cells fuels their growth, according to lab dish experiments in both papers. Multiple cancer cell lines proliferate more in the presence of neurons than they do on their own. In addition, synapse formation genes are upregulated in cancer cells grown in the presence of neurons, and neurons have more synapses and show increased activity in the presence of cancer cells, the US team showed, offering this and other evidence that metastatic cells adapt to the brain's environment. Chemicals that block nerve signals dampen cancer cell growth but do not extinguish it entirely, according to experiments from both teams. This indicates that paracrine signaling, which involves chemicals released into the extracellular space, contributes to neurons' growth-promoting effects, but that synaptic transmission plays the primary role. Really, that direct contact was needed, Vermeer says. So it really shows that it is a very intimate connection that is happening between the small cell lung cancer tumor cells and the neurons. Activating neurons in the promoter cortex in mice via optogenetics triggers increased proliferation of small cell lung cancer cells there. The US team further reported, showing that the neuronal signaling also promotes tumor growth in vivo. Conversely, withdrawing nerve signals may interrupt cancer growth. When the team severed the vagus nerve in a mouse model of small cell lung cancer before the animals had developed tumors, the mice ultimately developed fewer and smaller lung lesions. Spread to the liver was delayed or reverted altogether, and the mice survived longer than animals with an intact vagus nerve. Neuronal activity involving either the excitatory neurotransmitter glutamate or the inhibitory neurotransmitter gamma-aminobutyric acid GABA can drive the proliferation of small cell lung cancer cells, both teams found. Previous work focused mostly on excitatory transmission, but GABA has been shown to contribute to the growth of certain forms of pediatric brain cancer, says U.S. study investigator Michelle Monge, professor of pediatric neuro-oncology at Stanford University. Because small cell lung cancer is thought to arise from pulmonary neuroendocrine cells, which act as environmental sensors and communicate with pulmonary nerves, It is a good place to start looking for synaptic connections with neurons, Venkatesh says. But functional excitatory synapses also exist in brain metastases of breast and skin cancer. A third team of researchers reported in a preprint posted on bioRxiv last year. One of that preprint's investigators, Frank Winkler, shared this year's Brain Prize with Monge for their discoveries in cancer neuroscience. There's a lot of lessons to be learned from the neuroscience of primary brain cancers to be applied to what we see in brain metastases, Monge says. Unlike Monge and Venkatesh, Baleja is new to cancer neuroscience. His work began with a series of bioinformatics studies, including an unbiased, across-the-genome search showing that disrupting synaptic genes promotes small cell lung cancer in a mouse model, and a search that turned up pathogenic variants in such genes in human tumor samples. We were not looking for this, he says, citing that fact as a strength of the larger study that resulted. The data kind of slapped us in the face with it. Beyond basic discovery, the new findings open a completely new avenue for treatment of small cell lung cancer and perhaps other cancers, says Sebastian Talbot, Associate Professor of Biomedical and Molecular Sciences at Queen's University in Ontario, Canada, who was not involved in either study. In fact, a widely available anti-seizure drug that indirectly both enhances inhibitory signaling and diminishes excitatory signaling dampens the growth of small cell lung cancer tumors in the brains of mice, according to data in the U.S. team's paper. In the other analysis, two drugs, one experimental, the other in clinical use since the 1990s, that target a type of glutamate receptor showed some, albeit less dramatic, effects on lung tumors and survival in mice. Repurposing drugs already approved in psychiatry and neurology as part of cancer treatment would potentially allow a very quick translation to the clinic, Talbot says. This audio was created using artificial intelligence. Stay up to date with the latest neuroscience news and perspectives at thetransmitter.org. Follow Audio Research News on Spotify, YouTube, or wherever you get your podcasts.